Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Today's episode is personal, profound, and full of insights. I'm joined by Dr. Alex Serafini, an MD PhD trainee whose path into science began not with curiosity, but with pain. Chronic, unresolved pain following surgery led Alex to ask deeper questions about treatment, biology, and the very nature of suffering. In our conversation, he shares how his personal health journey ignited a research mission, one that now spans RGS proteins, GPCR signaling, and the search for better pain models. You'll hear about his work in the lab that uncovered spontaneous pain recovery in RGS4 knockout mice and why he believes the future of pain therapy may lie in building backwards, from patient to phenotype, not just from bench to bedside. And if you're looking to deepen your own knowledge, head to Terry's Corner right now, the only expert-led, on-demand pharmacology corner. We release 4 new courses every month and host regular Ask Me Anything sessions with Dr. Terry Kinakin himself. Learn more today at terrykinakin.com. You'll find all other relevant links, including our featured partner, Celtaris Research, in the episode description. And now, let's dive into this episode.
Hello, everyone. This is Yamina for Dr. GPCR. And today I'm very excited because I have with me Dr. Alex Serafini. Alex, welcome to the podcast.
Thank you very much.
Very excited to have you on. For those listening to this podcast, you might remember my episode with Dr. Mikel Garcia Marcos, and he mentioned Alex. I can't remember if he mentioned it before or after we stopped recording, but this episode has been in the works for quite some time now because of all the scheduling issues. But Alex, I'm very excited to have you on.
I'm very excited to be here. Thank you.
So let's start at the beginning. Could you please introduce yourself and tell us who you are and what is it that you do?
Yeah, so I'm Alex. I grew up in California originally. Ended up going to Hopkins for undergrad and kind of took a twist in the career trajectory I was going to take because I was diagnosed with a pilonidal cyst, which is like a lower back cyst that can get infected, and the corrective procedure for that tends to cause reinfections the way it's designed. So I ended up actually having persistent pain. I don't know if I would classify it as chronic pain for several years because of that repetitive surgical process. And so that actually got me very interested in research because, clearly with the opioid epidemic, I wasn't able to get any stronger pain medications. And so I did my master's degree at Hopkins as well with Mike Caterina, who worked on the TRPV1 receptor, was introduced to pain basic science, mostly in the peripheral nervous system. And then I joined Mount Sinai for the MD PhD program, where I was introduced to Venetia Zachariou and had the opportunity to work tangentially to GPCRs, more in the RGS side of things, and also predominantly in the central nervous system. So understanding how central nervous system mechanisms can mediate persistent pain and some of the affective comorbidities that can come along with that, like anxiety, depression, addiction, vulnerability. I finished my PhD with her and followed her to Boston. She's now the chair of pharmacology and physiology at BU. Did a postdoc with her, and now I'm back in med school again, wishing I had a pipette in my hand.
Well, do you think you're gonna have a lab once you've completed med school?
The goal hopefully is to do a true physician scientist track. Pain is a very exciting field, especially looking at it from like the psychiatric angle. So ideally having maybe 20% actual clinical patient facing time and mostly working in the lab. I'm particularly interested in model development. So seeing if we can bring preclinical models much closer to the actual human experience than what we're using now.
That's fantastic. And I actually invited Vanna on the podcast, but it was at a time where she actually moved the lab to BU. So the responsibilities increased and the invitation still sent. So I'm waiting.
I'll nudge her. I'll tell her that I did a primer episode for her.
That is awesome. That is awesome. And I'm sorry to hear about your pain issues and everything going on with your health. I wanted to ask, as a child, were you interested in science, or did you always know you wanted to be in science?
So it's a very interesting story. My father is involved in product management and cloud backup. I grew up in Silicon Valley, so not from that angle. And my mom was focused mostly on Hispanic marketing. I'm half Mexican. And it wasn't until I started getting a little bit older that my family shared with me that my grandfather in Mexico, who has more of an endocrinology background, was actually one of the researchers that did a lot of the preliminary research behind the mini pill, the contraceptive pill. He worked for a company called Syntex. It had a branch in Mexico for R&D. And he was able to develop the technology to a point that he was able to pretty drastically reduce an uncontrolled birth rate in Mexico with some of his approaches. And he's been acknowledged a lot for, what he's done. But every time I meet with him, especially when I was younger, it was mostly him trying to push me, like, this is what science can do for the world, even if you don't think you're going to be having an impact in the moment. And he didn't have any of his grandchildren that had gone into medicine or science yet. So he was really pushing. I was one of the younger grandchildren. So that kind of got me a little bit interested in it conceptually. But I would say that I didn't really start taking it seriously until I got into college and I started going through what I was going through.
Yeah. What an interesting story. So the interest into science jumped a generation.
Yes. Oh, yeah. A hard jump. No one in my mom's siblings was anywhere near the science realm.
Interesting. Yeah. Interesting. So how did you land in your PhD program, or I know what got you interested in research, but how did you pick your PhD program, PhD advisor?
So basically everything about what I've done is like the very non-canonical way to do things. So Mount Sinai is unique in that it has a program called FlexMed. And its intention is to try to bring people that are either highly skilled in, like biology, neuroscience, or to actually bring people from very hard sciences like engineering and computer science into medicine and grant an acceptance basically in your sophomore to junior year transition without taking the MCAT. For me, I was debating, I studied a lot of finance in undergrad too. I was debating, should I just not go into research and kind of just skip that or actually go for it? And Mount Sinai offered me this, which was a very great experience and also allowed me to study things that I was more interested in. I was still kind of hesitant. And so I ended up applying for jobs and was almost going to work in pharma. I had an offer. And then I ended up getting an acceptance on top of the MD FlexMed to the MD PhD program. And that to me was very exciting. And then on top of that, my parents were like, look, if you don't accept this, we're going to basically disown you. So they gave me the nudge I needed to really, take that step. It's an 8-year commitment, right? And it has its positives, but that's a long time considering residency is on the other end of that. So I never really had choices when it came to my PhD program just because of the nature of how it worked with Mount Sinai. The choice for Vana was also easy. Of course, she was really doing a lot of work with like bulk RNA-seq in pain back when that wasn't a super popular area, and also when people weren't really involved in looking at the genetics of CNS circuitry in pain. And she, at the time, Mount Sinai wasn't necessarily the strongest pain research environment. So she was like, target lock, that's the lab I'm going to go to. I interviewed with her for the PhD program as well. And all of that aligned very well. And I was grateful for the chance to work with her.
That is awesome. And then I'm curious, because before we hit record, you mentioned that you tangentially, and in the beginning too, worked on GPCRs. What type of work, or what were the questions you were trying to answer during your PhD and postdoc?
Yeah, so the reason I say tangentially is because I feel like I never was particularly explicitly interested in GPCR signaling. Vana 100% is. But the thing about GPCRs that I think everyone that listens to this would appreciate is you always kind of end up landing at a GPCR as a major target. And so one example, because I was interested in models, I worked with Dr. Carey Price on a paper where we tried creating a new model where you injure an animal and then you expose them to a high dose of oxycodone and then you withdraw them. And you look at how behaviorally they're worse. And you can end up doing sequencing for like various CNS regions that regulate pain. That's an example of like working on mu. And there's a lot of like downstream targets we didn't delve into too much because we liked focusing on epigenetics that were, you know, GPCR related targets and RGS related targets. Another example, which was not originally part of my thesis, but something that Vana was very interested in, and I agree it's a very interesting approach, is Vana has a wide array of RGS knockout lines, constitutive knockout mouse lines. And so working with John Javich at Columbia, we were able to get our hands on tianeptine, which is right now considered like a street drug. It's an atypical antidepressant that hits mu as well. It has some addiction liability, but it's like very effective and works more quickly than standard antidepressants we would think about in treating chronic pain. And so because the GPCR side of it, particularly on the opioid side of the signaling, was not super clear mechanistically, Vana had an idea, well, what if we, instead of starting with the molecular side of things, like use in vivo screening and looking at behavior to see how different RGS cascades can affect how this drug works. And so we would do like nerve injuries on animals and apply tianeptine and then see what the results were. And then go and do RNA sequencing on like specific animals that made sense. So that's an example of maybe like a very not traditional way to look at things. We also did some work in the peripheral nervous system with RGSs as well. That was more targeted. The lab is interested in RGS4. And we did some work during COVID as well, because that kind of interrupted my thesis. We decided that it would be interesting to work with a collaborator in microbiology, and I would get certified to go work in a BSL-3 lab and just harvest like dorsal root ganglia and brain regions, sequence them, see what the targets are, see why people have persistent pain after COVID, why some patients had chronic pain that went away in the acute phase of COVID, which has like some anesthetic translational potential. So really there's a lot of exploration, but because we use a lot of high throughput assays like RNA-seq, there are always like a plethora of GPCR targets and RGS targets that I didn't personally have enough time to investigate at a very deep level, but hopefully some work from my postdoc will come out soon that looks a little more deeply at one of the RGSs.
I love it. Thank you for that. And I think it's important as well to do this type of work in vivo. And anyone listening to the podcast knows that I'm very much experienced in vitro pharmacology, molecular pharmacology. But I think at the end of the day, especially when it comes to drug discovery, you can have the best looking molecule. If it doesn't do what it's supposed to do in vivo, then...
Yeah, and then especially in the pain field, even if it does what it's supposed to do in vivo, half the time it doesn't really translate to the clinic, unfortunately. But I think there's a lot of room for GPCRs to have their moment more in pain because we're using really outdated drugs in the field. I think there's so many more elegant targets that are a little closer to the GPCR side of things. They came out, but right now we're kind of focused on ionotropic targets. So like we have this big new NaV1.8 inhibitor that came out from Vertex recently. You know, it's not really getting the job done. We'll put it at that. And a lot of effort was invested in like targeting this one sodium channel, and it's expensive too, right? Like if you're targeting one specific mechanism, it doesn't, like, the downstream cascade is probably a little bit weaker than if you were hitting a GPCR. The length of action is shorter ionotropically.
Exactly.
The gene expression changes are not as robust. And honestly, like with the trials, they were not really that sufficiently better than opioids, right? And in itself, that's kind of a suggestion, you know, maybe that's hinting at something. But it looks like, interestingly, a lot of the orphan receptors, GPCRs, that people are looking at more tend to have a very interesting inflammatory component, like inflammation regulation, from what I've been reading in recent papers. And I think that will be very interesting because the pain field is really starting to understand how relevant different inflammatory cascades are to mediating acute pain, maybe the transition to chronic pain itself. And there's a lot of new tools coming out for that. And I think it's good timing to kind of take out the cheerleaders for GPCRs and say, hey, you know, this is another way to target this type of signaling.
Exactly. I love it. I love it. And I was thinking, you know, about timing and how long an inflammation pathway is activated and what does that mean for the cell? And does that mean that it's going to activate another GPC? Obviously, the cells express multiple receptors, multiple GPCRs, multiple other proteins. And I always find it fascinating to think about how much work a cell does have to do in order to not only perceive the information outside, but also integrate it and then response. And part of that integration also depends on how long you're getting that information and what happens to that information once it gets translated inside the cell.
Yeah, and I think particularly in the peripheral nervous system, like the dorsal root ganglia, that's a very interesting question, because you have a lot of players that you don't normally have in that region that can contribute to pain. So for example, you have Schwann cells that can kind of take this immune-esque phenotype depending on the damage state. You can have dorsal root ganglia neurons that not only release chemical factors that attract peripheral immune cells, but during COVID, it was interesting. We were trying to look at some different markers for immune cells, just with like IHC. And we had a hard time because the DRG neurons themselves would be labeled. And it wasn't non-specific. They would actually, they're almost like pseudo immune cells. They have these cascades activated that, you know, generally you might not expect in somatic cells to be active. So there's a lot going on there that we don't understand. It definitely makes sense as to why you can have these longer than expected immune cascades in a relatively privileged area of the body. But yeah, no, I think a lot of work is going into that right now.
It makes me it always makes me feel like we know so much, yet we don't know anything.
Yeah, well, especially in the pain field, I feel like every 5 years we're redefining our understanding of pain, right? So like a big debate now is, do people actually transition from acute to chronic pain, or is it a completely different underlying system that's working? And you have like questions like, if you had influenza 10 years ago, does that prime you to have chronic pain if you get an insult today that's minor? Yeah, I agree. I think that, you know, aside from the molecular understanding, we have even the conceptual framework, and many models and clinical phenotypes are still theories, very much so.
Once you complete your training, and you mentioned that you really would like to have your own research group, do you think you're going to be focused still on pain, or are there any scientific questions that you find intriguing at the moment that you might want to explore on your own?
Yeah, no, definitely. I think pain will always be a central component of, you know, knock on wood, if there is a research group in the future, of that research group. I think another very interesting thing I'm curious about is transgenerational signatures of pain. So, you know, like epigenetics is the sexy word of the hour in most fields. And I think that it's cool because you can have very broad signatures that are altered epigenetically, including GPCR signaling, that could be due to chronic pain in the mother or father. And depending on the parent, it might have a different effect. Depending on how the offspring is fed, it might have a different effect. If there's comorbid opioid use, it might have a different effect. So that's why I think model systems are very important before maybe starting in the dish, because there's so many variables that are really, really tightly controlled in a dish that might not reflect some of the variability in a living creature, particularly when we're talking cross-generational persistence of signatures. So that might be an alternative angle as well.
I find it fascinating, especially when it comes to animal models. Just not just, but the age of the model, of the animals, and the gender of the animals also affects the outcome of what you're doing. So if you're thinking about animal models and then you take it into humans, which are way more complex, because there is the changes in diet, exercise, genetics, where you live, how much you move, what you experienced while developing as a fetus.
Yes.
So like all of these things, and I find it very, well, obviously it's not easy to measure, but I find it fascinating that some of these events that happened to your system, to the body, actually determine, I'm pretty sure they determine some outcomes, but to what extent? And how do you prevent, or how do you bias towards a favorable outcome?
Yes, I agree completely. It's very funny you say that, because sex differences, particularly if you're looking at the inflammatory angle of pain, are highly relevant in the field right now. So for example, you look at spinal cord inflammation after pain, there have been studies that show very elegant cascades, that involve perineuronal nets and a very intricate cascade, and it might occur, like, let's say in males, very robust, not really, no concerns about it. And then you look at it in females and it's like completely irrelevant. We know from some of our opioid withdrawal work, particularly in the context of pain, that female mice are like pretty resilient when it comes to withdrawal states. And I think we don't really know clearly what that looks like in people. And I agree with you that I think, pharmacologically, especially if people start working on, more novel therapeutics and their GPCR based and everything, we don't know what it looks like person to person. Like a C57 line might have a sex difference in an inflammatory cascade, but honestly, probably there's a likelihood that in some women that exists and in some women it doesn't. And we have no idea what the dividing lines are. And if it's truly sex in mice, gender in humans, or if there's a spectrum of effects that maybe we're just looping into sex in mice that we're not really thinking about more deeply.
Lots of questions to try and answer, for sure. I think we went very theoretical out there.
Oh yeah.
And I know I asked this from everyone. I don't think it's relevant as we talked about it, but you mentioned, not necessarily having worked on a specific GPCR. GPCRs are always floating around in everything that we do. But was there any specific RGS protein that you preferred working with because of any given reason? I always ask this question, what's your favorite GPCR? But depending on the guest, I even got sometimes someone saying, well, actually they have a preferred or a favorite phosphorylation site, which I thought was really cool.
Yeah. So from an RGS perspective, when I first joined the lab, there was a master's student in the lab, Cleopatra Vrampu, and she had been working on RGS4. And there was this very interesting phenotype the lab found where, as a mouse in either inflammatory or like nerve ligation pain, was starting to enter what we consider the chronic pain range, which is about 3 weeks. the mice that were constitutively RGS4 knocked out started recovering spontaneously. And this is a recovery that is very rare to see at the preclinical level. In that study, they looked more, I believe, at like how specific thalamic circuits were affected and how that affected metabotropic glutamate receptor signaling. So that's a great tie to GPCRs. Unfortunately, I wasn't part of that project that much. But we decided to continue looking at RGS, and in some studies, RGS4 is a very powerful target, I would say, because it's expressed very robustly in the peripheral nervous system, which is probably the most powerful place you can intervene with some kind of therapeutic, but also in areas like the prefrontal cortex and thalamus that are very important in perception of pain, how your cognitive abilities might be altered in the context of chronic pain. So we decided to work, and this is some of my postdoc work, we've been looking more at RGS in the peripheral nervous system, and this is something that we worked on with Dr. Garcia. It is a very relevant target in the peripheral nervous system. I'll leave it at that because the work isn't out yet. But I think that the nice component to step back and speak a little more broadly about RGSs is they have a lot of non-canonical functions, right? They can serve as like scaffolds for different groups of proteins that might be almost completely isolated from the standard GPCR signaling pathway. And that's something Vana has spent a lot of time characterizing. And I think those arguably can be some of the more important aspects of the cascade to look at. And she's seen that with like RGSC1 before, that, you know, some of the non-canonical effects work very well with like mediating opioid effects in the context of addiction and pain. So my answer is RGS4. I have worked with many RGSs, like in the tianeptine study, we looked at RGS9 and RGSZ as well. And they really are interesting. If you could figure out a way to bias signaling towards one of the RGSs, you can end up having like radically different effects. So yeah.
I love it. I love it. And, you know, when I think about RGSs, or I think about beta-arrestins or other effector proteins that, you know, people would say, oh, you know, it does this, period. I don't think we have spent enough time or have the tools yet to better explore what other functions away from the plasma membrane or away from GPCRs that these proteins are involved in. And can we
Absolutely
target them?
Yeah, and you mentioned trafficking. I have not worked with trafficking all that much, but I remember going to one of the GPCR Gordon conferences and seeing some of the work that was done on where, you know, intracellular GPCR signaling, even when they're like an endocytose. I think it's going to be a game changer. I agree with you that, you know, we're kind of like locked in biasing with like one or two proteins, but the field is definitely starting to make the picture even messier in a great way with GPCRs. And, you know, RGSs are even messier than GPCRs, I think. So that's going to get probably even more messed up as well.
Absolutely. That's why there is an RGS and beta-arrestin and G protein meeting, separate, dedicated to these proteins. Not that we don't like them, but it's a whole world, whole other world out there. So let me ask you this, going back a little bit to your career. So you've completed your PhD and then you decided to do a postdoc in the same lab. Was it something that just came out naturally because you completed your PhD and you wanted to stay a little bit longer before going back to med school? Or were you considering maybe switching labs for a year or two before going back to med school?
Yeah, so I only did a postdoc for about a year and a half. And two reasons for that. One reason was that the MD PhD program was like, you cannot, you know, like take too much time off. So keep that in mind. The other reason, three reasons. The second reason is Vana was kind enough to allow me to pursue an MHA, a Master's in Healthcare Administration, while I was working in her lab. I didn't have to take as many courses in my PhD because of the MD PhD. So I think the administrative environment in clinical medicine is crazy, and understanding that more is better. That's not very relevant right now, though. The third reason is, honestly, working in Vanna's lab felt like I was working in like 3 different labs at the same time, and that she allowed me to work on quite a breadth of projects, which maybe you'd get a sense for based on the projects I was mentioning before. And basically, like, I just had too many projects to be on my thesis to walk away from that were incredibly promising, and I think, you know, highlighted some new mechanisms that will be very interesting to build out in the future. So, you know, I didn't feel like I didn't learn enough during my PhD assay wise. I really felt like I had a pretty wide arsenal after doing my PhD, and didn't feel like I needed to work somewhere else to gain some new skills. She really allowed me to also, during my postdoc year, learn a lot more about grant writing for R01s and a variety of academic administrative tasks that I think many people need to learn the hard way early on, and I kind of got a nice primer through this. So from that perspective, that was also very useful. It was like here's a crash course for 1.5 years, is like what it would be like to be a junior PI.
I love it. And I think and you made a great point. I think it's very important to be able to have this type of experience, because oftentimes during your PhD or let's say your regular postdoc experience or experiences, you have to produce papers. The end goal when you're in these programs is to produce papers, and you don't necessarily have the time, or maybe you're not in a lab where you get a chance to do all of these other activities, or you get the support. So whenever possible, taking advantage of this opportunity to learn what it looks like, what it feels like to be, you know, running your own group and writing grants and getting a sense of the administrative aspects of the job is really valuable.
Yeah, and I think another angle too that I learned in Vanna's lab is I've always been very jealous of people that are experts at one highly sophisticated method. So when I was in Mike Katerina's lab, that was in vivo DRG imaging, right? You can keep the animal alive and record a dorsal root ganglion cell or patch onto it while it's alive and being stimulated. That's a crazy technique. You know, many people dedicate sometimes even a decade to perfecting it. And Vanna did push me to learn harder techniques, but I think she also showed me the value of collaborating with other groups, and she was comfortable with me learning how to play a more managerial role in a lot of projects, as opposed to saying like, look, you are required to learn this very difficult technique for the lab, and that's like your goal during your PhD or postdoc. And so really a lot of the studies we've been able to do is because we've been able to be comfortable bringing other people's skill sets in and bringing in really whoever we needed to reach a finish line for an interesting project. And I think in many opportunities that's not available in many labs.
Yeah, no, I think it's true. And I wonder if it's because it's a different track. You know, in your case, and it's also, I think, a perception. When people know you're an MD PhD student, they think, oh, you know, you have a different profile or a different, let's say, conformation than your regular PhD or postdoc. Therefore, you might get more opportunities. I don't know. I'm just trying to get a sense of.
So another reason I think that this is relevant now is I think academia is becoming more collaborative, but also less collaborative at the same time. It seems like based on some of the grant situations I was involved in, it's at face value, everyone wants you to collaborate, but if you're at different institutions, unless you're submitting a U grant, like it's punished essentially to be working across institutions. So I think a lot of universities are adapting to that by really taking these people that know a hyper specialized skill set and turning them into a core and allowing them to really flourish and put their hands on different fields, but maybe not bear the same responsibility of having to be a lab that is constantly innovating and working on your own projects. We see this a lot at Mount Sinai. Mount Sinai is, I think when I joined, they were on like a nature list where they were one of the highest institutions for not needing to collaborate with other institutions. Like all the authors were within the hospital system. And they have really been pounding out a lot of cores for sequencing, nanoparticle delivery, machine learning for your projects. So I think ideally it would be, I think it's a very good skill set for scientists to also learn how to manage, given that I think most institutions, the allure for the few candidates that there are to, you know, become investigators is, what can I kind of like offload onto the cores and not have to hire a postdoc that that I'm competing with 20 other labs to hire because they're so specialized. So that might be another angle as well.
I agree with you. And it's an interesting thing. I think the academic system is so set in their ways. It's like a box, and this is what you can do, and period. And I think over the past decade, it has kind of become a little bit more flexible, but still we're not to that extent of flexibility that would allow to get to learn different things whenever you're a PhD student or you're a postdoc. You mentioned initially before going into med school that you got a job offer in a biotech or a pharma. Why not take that position?
Yeah, so like I mentioned, I studied finance when I was an undergrad, and the offer was actually at Roivant, which was started by politically relevant Vivek Ramaswamy. This is how he made his money, and I liked them. I don't think I would have liked the standard pharma job, but they had a very interesting business model where they in-licensed small molecules that had failed at meeting the indication endpoints at other pharmaceutical companies. So let's say Alzheimer's phase 3 fail, but phase 1 and maybe phase 2 were successful. So we had a good safety profile. And basically they would use various analysis strategies to predict that, you know, maybe this small molecule would be relevant in a different indication, come to an agreement and essentially recycle the small molecule and test it in different indications. And so it was really a way to do kind of what I've been talking about, like be more of a manager and take something that someone else has built and it didn't quite make it and make it successful. And for me, that was highly, highly alluring as a concept. And they've been successful with some of their attempts and very unsuccessful with others, but the overall success rate is higher than if you're doing a full analysis, a full development strategy within your own company. Honestly, the only thing that pulled me away from it was the MD PhD program. I personally, I could never bring myself to do the PhD by itself. Not obviously because there's anything wrong with that, but I just felt that the process, I didn't feel like I would be in a position after my PhD where I would feel like very safe. I was hearing so many instances where people were having a really hard time finding jobs, and that some pharmaceutical companies were leaning towards PhDs being overqualified and asking a lot for master's degrees or bachelor's degrees to pay them less. So I don't know, I think I was afraid of trying to just do the PhD by itself. And I was also afraid of the MD by itself because there wasn't, it's a lot of regurgitation. And innovation can be very difficult in medicine until you're a senior physician, for the most part. So that's where pharma kind of bridged the two. If I could make my way in, then I could take care of all of those concerns. It's safe. I can innovate. I could learn about different indications on the business side of things. But I think the MD PhD for setting myself up long-term to be successful was, in my opinion, the better route.
And also it gave you the satisfaction of, one, having a direct impact on patients, but at the same time staying close to research.
Yes. Yeah. It's very interesting. I think I think a surprisingly low number of people in MD PhD programs become physician scientists, either because they really love being patient facing or they absolutely hate it. And the risk with medicine is that there's a very high burnout rate, and sometimes that can detract from you actually doing anything on the science side. But yes, I think being able to be more intimately knowledgeable about a patient's condition can really guide your science and what are really relevant experiments to do versus like fluff experiments, let's say.
Yeah. No, I agree. I love it. All right. So, top 3 aha moments that you had that shaped your trajectory. I think in the beginning you mentioned the ultimatum. I don't think it was a suggestion from your parents. It was more of an ultimatum to make a decision.
Yeah, they were the backup in case it wasn't clear to me. They would make it clear. No, I think they would have respected either way, but they let their opinion be no. Yeah. My first aha moment after that was in Mike Caterina's lab. I had never worked with animals before, and he was probably the best mentor to learn how to do that from. Incredibly humble, and he just kind of like has a great knowledge of everything that's going on. And I remember I was being trained on the spared nerve injury surgery, and I spent like weeks not getting it right, and he was always there to support. But the main thing I remembered was I was deathly afraid of gripping mice. And here he is, like he's in R01 season, and I would go up to him, I'm like, I'm just not sure I can do this. So he got up, he has this kind of like devilish smile where you know that you probably shouldn't have gone to him to ask him something. And he found the fattest, meanest C57 that he could find, and he just stood there and looked at the box and looked at me and said, we're going to do this. And he stood there the whole time until I did it, despite all of the deadlines he had. And he taught me a lot of lessons. And I think he taught me, that was the first time I could see myself being a PI and making it all the way and understanding what it's like to have a support system. So working in his lab was the aha that kept me interested in research, period. The second aha moment was when I was working in Vana's lab, and we were working on COVID, and some of the first reports of people having persistent pain came out, and we just went in and we're like somehow going to take DRGs from hamsters without a microscope. So you can't really have a microscope because everything's in a hood. DRGs are like super, super tiny in rodents. So here I was like trying to pick them by eye, and we just took them from a hamster and sequenced them, thinking honestly probably nothing was going to happen. And we just saw the most robust gene expression signature from these hamsters in the DRG. And it was so persistent. And that was an aha moment for me, especially working in the CNS where pain signatures can be like pretty nuanced sometimes, that like it's good to just keep asking questions and pursuing them. And probably a lot of them are not going to work out, but it's not wrong to like fish a little bit, like start a project with an RNA-seq on a model and just see where it goes before kind of like doing the true NIH strategy of hypothesis, then we'll spend the money on sequencing. Yeah. Hopefully no one at the NIH ever sees this in case I ever ask for funding. And the third aha moment was again from like a mentor side of things, that if you're with Vana, if your PI really, PIs have the power to really prepare their students, even at just the grad student level, right? To see what it's like to be a PI. I think F grants are like an interesting start, but you don't really get like a full peek under the hood. And Vana really gave me the opportunity to see like, this is how much it sucks to write an R01, and like get involved in this, see what it's like, this is what it's like to work with another institution and get the MTA set up and the financial connections set up and everything. And for me, it was an aha, like, okay, I can finally see what this is like. And I felt comfortable because I wasn't completely scared of it. I was like, okay, this is doable. And I know if I go in the future to be a PI, I have PIs from my past that are willing to like share previous grants and help me get where I need to be.
Yeah, thank you for that. I think the grant writing aspect, the sooner you get a taste of it, the better, because that's what's going to determine whether you really want to be a PI or you just want to run away from academia.
Yeah, I agree. And it's unfortunate because I think a lot of people don't have the experience to see that until they're in it, in the moment. And it can really sideline you on your path to success if it's not the right fit.
Exactly. It's not about pipetting. It's not about learning to run experiments. It's all about being able to enjoy that kind of writing, because it's a very specific talent. It's not like writing a paper. And
not at all.
I can definitely tell you that I do not like to do that. It is not something I particularly enjoy. I applied for scholarships, and it was very painful to go through all those forms, and you have to be next level organized, and that's just the administration of the paperwork. And I'm not even talking about the science, the hypotheses, all the reading, all the papers, and everything that you have to put into that application.
Yeah, I agree. The one thing I actually was surprised that I liked about grants is I feel like when you're writing papers, it's not completely acceptable to write your entire thought process out into the paper. Like, I ran this test because of this and did this because of that. You kind of have to keep it tight and readable. Yeah. But the grant, you basically you're explaining every step, why you're doing it, what statistic tests you're using, why you're using it. And I think it's a great opportunity to really put all of your thoughts on how you run a project in a lab onto paper. And it's definitely painful. It's like very detailed and meticulous, but I kind of enjoyed that about it.
It's interesting that you mentioned that. So very recently, I got invited to teach a course on strategic project management at UNC Chapel Hill. I want to say also that I'm very grateful for Dr. Kinakin for inviting me. When Terry calls and says, I need you to teach this, you say, yes, sir, and you go and teach it. And it was a drug discovery course. But actually, the concepts that I ended up putting into that deck, and the major take-home message was build backwards. Figure out what's your end goal, and then build backwards. What do you need? Who do you need? How are you going to get there? And that's what's going to allow you to also plan out these things. And I think just as much as important in drug discovery, it's also important in academia as well.
Yep, I agree. And I think harping back on the MD PhD discussion that we had, it is a privilege because you can go into a project and your end goal is seeing like a patient that is in irreversible pain receiving an infusion, and like, what does this drug need to look like?
Exactly.
How is it going to be delivered? How long does it need to last? How is the patient going to react to it? And you might not think like that's maybe like way too in the future, but it starts kind of slowly providing rails for how you need to be thinking about the discoveries that you're working on. But I agree. And that's, I think, kind of the same logic that Vana brought with this in vivo RGS strategy, right? Is you're kind of, from an experimental perspective, not from like a project management perspective, but starting from the end and working to a more refined, like a top-down approach, let's say, right? Makes sense in a lot of circumstances. And I think it's taught to not make sense. And definitely the bottom up makes a lot of sense in science. Clearly, starting from cells has a lot of advantages. Yeah. But maybe we can be a little more open about fishing or starting from a top-down approach sometimes.
Agreed. I love it. All right. Any advice for scientists or for anyone who might want to join an MD PhD program or think about, this type of career where you're actually marrying seeing patients, but also working with animal models and with model systems?
I think my advice, MD, PhD or not, is to always at least superficially learn as much as possible. And again, I think this is something that maybe is kind of like, it's treated as a bad thing to do in like a specialized graduate degree. But beyond the science, right? Like if you're doing medicine, spend a lot of time with patients, interacting with them. When you talk to patients that have an unmet need, you learn things that no one is thinking about in terms of how to solve those problems. Interact with your tech transfer office. Patents are something that people don't talk about in grad school, but in many institutions, that can be a make or break. Ask your PI before you join the lab, will you let me work on high stakes grants or projects, and is making mistakes okay if I want to learn about a bunch of different things? So I think it's always important to be very, very skilled at specialized things. But I think during this time in graduate school where you have the flexibility to discover and the ability to be forgiven for making mistakes, you should learn as much as possible about the whole system.
I agree. And unfortunately, not whenever you go into these situations, grad school, or even a postdoc, you might not necessarily be focused on these types of goals for yourself. You're mainly focused on experiments, hypotheses, writing papers, and moving on. But it's a package deal.
Yeah. And some people want to be very focused as well. Some people don't have the opportunity to be a bit broader, and some people don't want to be. And I think at the end of the day, that is totally fine. We need everyone under the umbrella that has different perspectives. But I think personally speaking, I receive a lot of joy kind of being able to see the full big picture of how things work and where I'm currently fitting into that puzzle.
Yeah. And I think it's also motivating, although everyone might want to work on the big picture, but they want to work on, I don't know, a phosphorylation motif. At the same time, it's important to put that into perspective.
Yes, exactly.
To understand, why am I working on this particular, and I had the phosphorylation motif was just an example. Why am I working on this specific protein? Why am I working with this model? And that requires the ability to look up from the bench.
I agree. And I think even one step further, one thing that we think about all the time but don't really think about is, there's a very high preoccupation with what journal am I going to publish in, how many citations is it going to get. But you don't think about, like, all of those citations are other people watching your work, right? And other people taking your work and adapting it in a different way, or taking it one step further in your field, and understanding the big picture of how someone else can take what you're doing and maybe commercialize it, do something else with it. That can really guide, I think, not only what you're doing, but why you're doing it. I remember when I first, and I think every grad student has this, like when I got my first couple of citations for my papers, I was like, oh my God, like I never thought this day would come. But the first thing I thought of was I started clicking on the citation number to see who was publishing this. And I loved reading like what other people were saying about the paper. If it wasn't just like chunked in the intro as like a 20 citation for one sentence deal.
Exactly.
But it made me think for all of my future studies, like here's where this might go. Personally, maybe it's going to become a patent, but for other people, maybe a grad student won't have to do this experiment or can skip this part of their thesis because I helped answer a question that they were curious about. Or they can use my data to make their lives more easy for like sequencing. So yeah, perspective I think matters a lot. And I think many people don't think about it, like what perspective really means deep, like at a detailed point of view.
No, I agree. No, I agree with you. And to your point, you mentioned, looking at who's citing your paper. I always enjoyed looking at that as well, because sometimes my paper gets cited and I'm like, wow, why are you even citing my paper? Then I dig deeper. I'm like, oh, okay, now I understand. But sometimes those citations come from a field that not necessarily focused on, the molecular pharmacology of that receptor I published about, but it's something adjacent. And
Yeah.
they use that information to then build out their program or their project.
And that can even be its own aha moment, right? Like, I didn't even think that my work could do this. And
Yep.
I just have to keep my mind open, you know? Yeah. So
Yeah,
agreed.
no, I agree. The biggest surprise / honor, again, and I don't know why this is the example that comes into mind. So the other day I got an email from Dr. Kanakin again, who says I think the subject was your fame precedes you. And he messaged me and he's like, oh, I actually didn't realize that it was you who was the first author on this 2-author paper. And I cited your paper in my review a couple of years ago. And I was like, wow, what an honor. 2, you never know what that work contributes to the high level base knowledge of a specific field.
That's a really good question. I think I have the problem of being so anxiously productive that when those moments happen, sometimes maybe I don't pay attention or take it in as much as I can. I think, for example, little things like you mentioned, Dr. Garcia suggested that I come here. And, for me, that's like a very heartwarming thing. I look back on my time with Dr. Katerina, and we were having like coffee one day, and he's like, yeah, there's probably never going to be another Alex in my lab. I think those little things are, for me, heartwarming. But I have this thing where I don't know, I like never really am good at absorbing honors that others give me. And I think it's something that you see a lot in like medical school as well, where people are kind of just like grinding as quickly as possible. Someone gives you a compliment or an honor, you're like, okay, thanks. Like, I have to go work on this paper now or study for STEP or something. Really appreciate it.
No, I agree. And I wonder if it's about this need of grinding and moving things forward, or is it the PhD or a scientist type of profile? It's at least with me, it's, oh, I reached this goal. This is great. What's next?
Yeah.
And it's hard. And I think it's important to stop and celebrate these little wins.
I agree completely. I think I tried at one point to do the champagne cork thing, where every time you pop a champagne to celebrate, you write down what the celebration was for, to try to have that, like for my thesis events and stuff. But I wasn't able to keep it up. So I think I'm also a bit young, so I'm gonna save like the honor, like something the threshold for really considering something an honor high in case something ever happens in the future, but with the understanding that it's entirely possible that nothing does.
Little wins. I think it's an accumulation of little wins. Yeah. All right, last question, Alex. What should we title this episode? I think that's the toughest question of the entire.
I knew that question ahead of time, and I've thought about it, and I still wasn't able to really land on it. Maybe something along the lines of like a different perspective on science. Because I feel like generally when I talk to people, they will agree with like a small part of my perspective on where I would like science in the US and the world to go, but never like the full picture. And I think part of that is also because I'm still a young, naive person, and I haven't been ground down by the way the system works, but at least for now.
I like that. I think it might be missing something. So it's very hard to title an episode before you had a conversation. And I think what came through is, one, the scientific gene skipping a generation, or the research gene skipping a generation, which I think could be a very interesting title. But I think the way you were talking about, going from a model system and understanding what happens in the model system, and then drilling it down to the MOA instead of doing the opposite, from dish to model or from model to dish, which could
maybe go with the former one, because I think when I nudged Vana to come on here, like that was her idea. So I think
Okay.
she could give a lot more details on that, and that might be a title best reserved for her. I definitely think, you know, being raised without immediate scientists in my generation, or from my parents' perspective, did play a big role, because I wasn't looking at research from the very beginning. And I know a lot of researchers in the pain field that are very prominent now was the same situation. And they come with pretty unusual but highly elegant ideas, these prominent researchers, and you can tell that they have a not as rigid of a mindset. So I think that would be an appropriate title for sure.
Awesome. Yeah, I called it Research the Gene That Skipped a Generation.
I love it.
Let's keep that, and then we'll reserve the model to dish title to Vanna.
Yes.
Although I think it also depends on what transpires in the conversation. Sometimes we never, with some guests, we don't talk science at all. We talk leadership, which is the whole other topic, very important, but different than direct, focused on specific science.
Makes sense. Just in case.
Just exactly. Alex, thank you so much for your time. I really appreciate it. Don't go anywhere. I'm going to stop recording, but I really enjoyed having a conversation with you. Very interesting.
Thank you for having me.
Huge thanks to Dr. Alex Serafini for joining us today. One moment that really stayed with me was when Alex described how a failed surgery and a lack of options pushed him to pursue science. It's a reminder that some of the best research questions don't always come from textbooks. They come from lived experience. If this episode resonated with you, send it to a colleague, perhaps a colleague working on pain research or translational pharmacology. To stay connected and amplify more GPCR voices, subscribe to the Dr. GPCR podcast and consider leaving us a 5-star review on Apple Podcast. Visit terrykinakin.com to explore Terry's Corner today and level up your pharmacology skills. Also, check out this episode's description for links to our partner, Saltaris Research, their fluorescent ligand tools, and their applications. As always, thank you to the entire Dr. GPCR team for making this episode possible. Because receptors don't work alone, and neither should you. Until next time, stay safe.